The promulgation of research is always dependent on the sources of the data and also media. That's my deep conviction. I'm, as a researcher, very convinced of is that knowing the history of biological experiments is one of the most important topics. Stem cell therapies have really gained great interest in all sorts of media and also in research. However, if you look really back into history, we are aware in Germany that in the beginning of the 30th and then the 40th of this last century, there was one prominent doctor, he was Swiss. He also promulgated already the idea that the injection of stem cell compartments has some importance and increases well-being of diseased patients. This doctor is called Professor Nihans, and he was working in Switzerland. So what did Dr. Nihans do? Dr. Nihans had a private clinic in Switzerland, and he injected xenogeneic protein compartments of different organs of different animals into humans, and was able to show, just with very few experimental data and much less human trial data, that these patients were doing much better after having received these protein compartments, these cell compartments. Here comes the second name I think is of great importance, is Professor Pischinger. In 45-46, Professor Pischinger, Austrian, originating from Upper Austria and also working then in Graz, went to Switzerland, working together with Professor Nihans and discussing about how can we stabilize protein compartments that can be injected into humans. And here at that time, Pischinger was giving Nihans the idea, because prior to that Nihans was injecting living cells, sterile living cell compartments into the bodies of diseased patients, and then Professor Pischinger came up with the idea to lyophilize these protein cell compartments. This idea was promulgated by those two, and basically they were very, very successful. They had their own clinic. And in the late 40s, Pischinger came back to Austria and was the first scientist who actually claimed that introducing xenogeneic protein cell compartments does harm to patients. Just to name you this technique, it's called the so-called Frischzellentherapie. Pischinger so claimed, and also that these reports were all published in the Wiener Klinische Wochenzeitschrift, claimed that introducing xenogeneic protein compartments, cell compartments, does harm because known risk for thrombosis, zoonosis are present. So this meant that he was really abstaining from this idea, but Nihans continued with his idea, and the Frischzellentherapie was basically something that was continued far in the 80s, and then actually I think in 86 it was abducted. As late as 80s it was abducted in Germany. So saying this, I want to say that introducing xenogeneic protein cell compartments is not a new idea. So in the late 90s, of course, researchers then found that stem cells can home to different cell compartments, and then really a great avalanche originated when in 2001 two publications published in Nature Medicine have shown, and that was really great scientific work, have shown that stem cells can attenuate myocardial infarction. This was then at that moment a complete surprise to the whole scientific world, and lots of groups ensued looking at the data, looking at the experiments, and then jumping from pure experimental settings in the murine, in the rodent model, into humans. And here I have to just allude to Professor Stauer from Düsseldorf. He was the first who actually utilized stem cells intracoronarily. He was a professor for cardiology in Düsseldorf, and he infused stem cells and was able to show that these hearts were doing much better. In consecutive years, Professor Drexler and Professor Wollert, they published a boost study where they were able to show that stem cell therapy augmented heart function as compared to control. So it was shown in lots of meta-analysis studies that stem cells do some benefit in the indication of myocardial infarction. It is of course of great importance to note that if you look at the papers directly, very meticulous look at the control experiments, these experiments claiming that stem cells are doing good in experimental myocardial infarction, something is missing. There were no control experiments. So just alluding to the Anversa paper, Donald Orlick paper in Nature, they just injected c-KIT positive stem cells and were able to show that these cells these hearts did much better than those that received saline control. So that means they were claiming stem cell is doing good, but basically there was no control experiment performed. In 2005, there was a very prominent paper published by Necki and Zhao. Those two researchers, Duke University researchers, have found basically or put forward the first time that if you see stem cell therapy, not the stem cell is preventing myocardial infarction, but the paracrine factors. That was quite a surprise. It was published in Nature Medicine and was cited till now about 700 to 800 times. So this was a formative paper showing that that stem cells have an effect, but it's not the stem cells but the paracrine factors of stem cells. In the same year, there was also a very interesting hypothesis paper without data, and published by Anker and Tum in the JAC, where they basically have shown that they speculated that basically the effect we see in patients who have myocardial infarction and who have received stem cells, that these stem cells might already undergo apoptosis because you take the autologous stem cells out of the body, you manipulate them up to 24 hours and then reinfuse via coronary route. And they were speculating that these stem cells are already undergoing apoptosis, and these apoptotic stem cells are attenuating the immune system, and therefore we have the effect seen. So at one side you have got the paracrine hypothesis, on the other side you have this Thun-Anker hypothesis that the apoptotic white blood cells are attenuating the immune activity in the inflamed heart after AMI. So these were the two really pivotal landmark notions I perceive as very important. This gave, of course, our group in Vienna the possibility to do the exact control experiments. So we looked at the papers, found that the mononuclear cells as control experiments were missing, and what we then did, we just took white blood cells of a rat, induced apoptosis in these rat white blood cells, and infused these white blood cell suspensions into a rat that underwent myocardial infarction, and have seen clearly that there was no infarct. I just want to show you this here. What is the notion now in the industry? The notion now in the industry is the following, that people just discuss, or industry companies just discuss various topics which is pertaining to biobanking, gene therapy, cell-based therapies, small molecules, tissue engineering and biologics. But actually something is completely missing, and this is from the Alliance of Regenerative Medicine. This was just copy and pasted from the present internet appearance. And actually what is missing in the whole discussion is that there's no secretome-based regeneration discussed at all. There's no discussion. All companies, all researchers still discuss about, yes, we utilize stem cells, we utilize cellular compartments. These cellular compartments might induce paracrine factors, but basically that the sole application of paracrine factors can have a role is not on the agenda at the moment. So what I I think that the industry completely turns a blind eye, and there's a complete cell-centric vision in the field. So just to rehash my notions from my first discussion, we took mononuclear cells. So mononuclear cells are T cells, B cells, NK cells, and monocytes. We brought them into apoptosis, and here we have got 4 microscopies of such a cell in a time-dependent way. So take the T cell out of the body, you have got the A, you've got a nice-looking T cell. If you induce apoptosis, you see a fragmentation in D, and that's after 24 hours. So seeing this, I want to allude to data where we've shown that that dialysis patients, artificial heart patients, septic patients have lots of apoptotic T cells, therefore they have no immune system. Of course, there was also another group in France, SAS, and they have claimed that apoptotic leukocytes can be utilized as anti-rejection therapy. That was by SAS in France published, and basically what we have found is that cells undergoing apoptosis, can be perceived as a bioreactor. So how did we basically make this finding? We took white blood cells of rats, induced apoptosis. We waited for 24 hours until we knew that these cells are already in the process of apoptosis, and then we induced infarction and applied these suspensions, the apoptotic T cell suspensions, to hearts and to rats undergoing myocardial infarction. And that was the result, and what we have seen, that the hearts had no infarct at all. So what is now the explanation for that? You can either say it's the paracrine factors or the apoptotic cells. I completely abrogated this idea, and the beauty of this idea is that you just take the paracrine factors and then you have got a lyophilized powder. This is the medication that we utilize. So the point being is that we had this great data showing that stem cells are attenuating AMI. We have the great data that paracrine factors of stem cells are attenuating AMI. We have the great data that apoptotic stem cells might immune modulate. And here all the 3 elements are now concluded in this one experiment. So we took now this paracrine factors, and we then utilized that in multiple indications. The first indication was in small and large animal, where we have shown that paracrine factors derived from white blood cells of pigs attenuates myocardial infarction. This was published in 2011. Then we have shown in an experimental model that paracrine factors of mice can attenuate experimental myocarditis. It means that it induces immune modulation. Third, we have shown that if you take these paracrine factors, put them into emulsions, and then treat animals having undergone wounding models, then you can attenuate inflammation and augment the healing of the epithermal layer. This is published already in 2013, and it can be depicted here. And last but not least, it was a recent publication where we even could show that if you applied paracrine factors and intraperitoneal application of these paracrine factors, we were able to show that stroke volume was decreased. This was published in 2014. So the next step was we went quite straightforward. We identified a GMP facility. This GMP facility is located in Linz, and we validated the whole entire process of paracrine factors derived from white blood cells. And the Austrian Health Authority mandated us to produce this medication, this medicinal product. It's termed biological, and therefore we can now start with first clinical trials. The first, and this is very important to me and to our team, the first paracrine-derived regenerative medicine trial commenced in October 2014, and where we sent first patients to Linz. We get the APOSEC produced, then we punch the patients, and then for 7 consecutive days they will receive APOSEC or medium control as a treatment. This entire work effort was supported by the Christian Doppel Laboratory at a university-based financing model by the Ministry of Science in Austria, respectively private funding of a company that is termed APOSEC. In conclusion, I think that I interpret the last 10 years that there is a change of paradigm has occurred. We know, I've alluded to work in the 30th, in the last century, where Nihans introduced xenogenic cell compartments to augment well-being of patients in need. He documented that just in anecdotal ways. There was no culture of clinical phase 1, 2, 3 trials. Basically, you can term this voodoo. Second, it was a very discussed professor, Professor Pischinger, who was the person who actually suggested lyophilization of these protein compartments, cell compartments, for getting these drug substance transferred to various locations in the world and for application. There were some islands where this treatment was then given. It is even interesting to know that the Pope was treated by such a therapy. Then there was the next phase in the 90s, where the first time it was described that stem cells are homing to injured locations. This was published very extensively in very nice publications. And then these landmark papers in 2001, which were really the first publications that showed if you take white blood cells, mononuclear-like stem cells, that these cells, when infused, can attenuate, or injected can attenuate myocardial infarction. Then 4 clinical trials have evidence that there is some benefit of stem cell therapy in patients having myocardial infarction. However, these were described to be rather meek, and I think the next augmentation of knowledge was then the speculation that not the stem cell per se are doing the good to the patient, but paracrine factors. This is really work originated by Gnecchi. And then in 2009, when we as group have published that apoptotic white blood cells are able to attenuate myocardial infarction. That was 2009, and in consecutive years it was also the proof that not paracrine factors of stem cells, but paracrine factors of white blood cells are doing good to the patient. We have shown in multiple indications that paracrine factors derived from white blood cells are doing good. We have identified a GMP facility that produces this, and last we have also identified multiple mechanisms that are responsible for all these effects seen and also claimed by stem cell secretome. What are these mechanisms? It's immune modulation, it's cytoprotection, it's anti-aggregation, it's vasodilation. So we have a pleiotropic effect, and these effects are due to a multiplicity of substances that are present in cultured media derived from white blood cells. So I want to allude to protein compartments, I want to allude to exosomes, I want to allude to phospholipids, to cholesterins. So we have a pleiotropic mix that is present in our drug product, which we call APOSEC. It's called apoptotic secretome, APOSEC. Last, I also really want to give credit to Professor Pischinger. He's an Austrian histology professor who basically claimed that already in the 50s of the last century that white blood cells are constantly undergoing programmed cell death and are secreting paracrine factors, and thus gives some sort, as he interpreted in his notion, as giving life energy. So looking at all the effects our paracrine factors do in patients who are in harm or preclinical harm, because we just have shown it till now in preclinical experiments, I would suggest that his speculation was right at that moment. Last, this whole entire work was financed by private investors, angel investors. The production of APOSEC, of our paracrine factors derived from white blood cells, is patented. So we have got the rights on the production of this compound, and we are looking forward in the next years to get this product to the market for the patients. I think this is the complete story. Thank you for listening.